Hello students and welcome to my video on cellular respiration. This is going to be slightly different than my other videos because I'm licensing it under the Creative Commons attribution, non-commercial license, which means anybody can use it, and I use no copyrighted images. So this is free to distribute to anyone in the world to hopefully help them understand cellular respiration. It just can't be used for commercial profit. All right, you've probably seen it many times. The mitochondria is the powerhouse of the cell. How come? Why so? Hopefully, by the end of this, you should be able to explain it to your family and friends. Okay, hopefully you remember from class that the point of cellular respiration is to make ATP. And what's the point of ATP? ATP is energy, right? And this energy lets us do stuff. So here are my kids this weekend, and they were full of it. A lot of ATP going on here. So let's dig in. Here is a nice picture. And we looked at it in class, and it's showing glucose getting converted in a series of steps, to ATP. Why can't we just use glucose as a energy source? It'd be like using a gallon of gasoline to try and light a birthday candle. It'd be too much. So we need to break it down in a series of steps to little bits of energy that we can use. You can also think about it like currency. So this would be like, 1,000 real note. Reals, for those of you not in Oman, is our form of currency. And this would be like single real notes. And so this would be like a single dollar for those of us from America. And this would be like a $300 bill that couldn't be exchanged easily. So every molecule of glucose is going to go through what's called glycolysis. Then you'll see if there's oxygen present, it's going to go through to the mitochondria. It's going to do what's called the Krebs cycle. Here it's abbreviated as the citric acid cycle, the electron transport chain, and make a bunch of ATP. No oxygen present, then the products of glycolysis, this pyruvate, is going to go through fermentation. This picture can be a little intimidating, but it's not too bad, and we're going to go through it, and we're going to figure it out. So let's do it here. All right. With this model, can you describe glycolysis, the Krebs cycle, and the electron transport chain? If you can do it off of this blank mitochondria, then you're really on point, and you should buy yourself a mitochondria t-shirt. I have one. If you can't, it's okay. That's tough. So let's break it down and let's go through it together. But go ahead and pause the video here and see what you remember. Okay, here we go. Let's do glycolysis first. Glycolysis, lysis means to break, and glyco stood for glycogen or glucose. So we're going to break the glucose. If here's my glucose molecule, we're going to break it in half, and we're going to make what's called pyruvate. I'm just going to abbreviate it as PYR. So here we have this pyruvate. We're going to have two of them. At the same time, we're also going to end up making 2 ATP and 2 NADHs. Well, ATP we know is energy. That's good. What is NADH? NADH, students, is like a taxi cab. And that taxi cab, when it's in the form of NADH, means it has high energy electrons in it. So it's going to take those high energy electrons to the party in the electron transport chain, which we'll get to in a little bit. So those pyruvates, what are those? So those are the broken in half glucose molecule. They still have a lot of energy within them. And so we're going to strip them into more taxi cabs in the Krebs cycle. The 2 ATP, let's review that real quick. It's going to take 2 to break the glucose. So we put 2 on there to break it. It's going to eventually give off 4. So if it took 2 in and it gives 4 out, the net gain is 2 overall. 4 minus 2 for 2. All right, let's go on to the Krebs cycle. The Krebs cycle is going to make 8 NADHs, 2 FADH2s. We already learned that NADH is a taxi cab. Think of FADH2 as another taxi cab. It just enters the electron transport chain at a slightly different protein. So if you're familiar with it, this could be your Uber and this could be your Lyft. It's just a way to carry high energy electrons. The Krebs cycle also makes 2 ATP. So far, for one molecule of glucose, we've made 4 ATP. And look at this. It makes carbon dioxide. And carbon dioxide, as you know, is what we breathe out. So you're kind of starting to see how the products of cellular respiration are the reactants of photosynthesis. And for that, you can go back to our last unit to review. The Krebs cycle takes place here in the matrix in the mitochondria. It involves a lot of enzymes to change these pyruvates into these products. So the reactants of the Krebs cycle are pyruvates, and it's going to be a little bit of ADP to make this ATP, and a bunch of enzymes. And what we get out are 10 taxicabs, 8 of the NADH variety and 2 of the FADH2 variety. If you remember in your POGL, you look through and you saw that the NADHs are going to make a lot of ATP. So are the FADH2s. So let's move on. So glycolysis, we split it. Krebs cycle, we used enzymes to take that pyruvate and make these. This is the big point. We made 10 NADHs and 2 FADH2s. And here comes the electron transport chain. From that, look at this, students. We're going to get a big bang for the buck. We're going to make over 30 ATP. We discussed in class that the numbers up for debate a little bit, and that's pretty cool, right? It's showing science is a process, that not everything's figured out precisely. The older books will say 38 ATP, but that's with maximum yield. So I guess that would be 34, right? Because you have your 2 here and 2 here, but that's under absolutely perfect conditions, and that's not always the case. Let's get to what's important, right? Is it makes a lot, and it makes a lot more than these 2. So we want to do the electron transport chain. And then look, it sends back the empty taxicabs. These are the full taxicabs, NADH. Here are the empty taxicabs, NAD plus and FAD. When you take chemistry and if you take AP biology, these are the oxidized forms of these, and these are the reduced ones. Okay, excuse me. So let's get a little bit into the electron transport chain. Notice also that it makes water, right? So now we're getting some of those reactants for photosynthesis. We got carbon dioxide and water. Now I'm going to erase these really quickly. I got to learn my gimmicks of how to super erase. We're running a little short on time here. And let's get into the electron transport chain a little more. There's a fun word to describe it that you can use at home called chemiosmosis. Oops. Here we go. Can you describe what's going on here? Here is chemiosmosis. So go ahead and stop it and see if you can figure out what's going on. All right. Hopefully this picture helped you out. I was glad I found this on the free interwebs. Here's the electron transport chain. There's 4 different proteins. Proteins 1, 3, and 4 are going to really be able to pump out these H pluses. So here comes our full taxicab. The taxicab is going to drop off 2 high energy electrons. These electrons are going to go through the chain. Each time they go through a protein, they're going to pump out an H plus. This is a proton. It's a hydrogen ion. It's just simply 1 proton. It pumps them out here into the intermembrane space. I'm going to go back a couple of slides. The intermembrane space will be here. Here's the matrix and an intermembrane between 2 membranes. So here, the H pluses accumulate. Those H pluses came from the H in NADH and FADH2, ultimately came from the H's in glucose. It's the way you can think about it. These H pluses get kicked here. There's more of them here than there are here. So that is a concentration gradient. It's also an electrochemical gradient, and it's going to diffuse. They're going to diffuse through what's called ATP synthase, my favorite enzyme. Oh, my head's covering it up. But ATP synthase is going to use this energy, this potential energy, these hydrogen protons flowing downstream to turn a rotor and attach a phosphate to ADP. It attaches it to ADP and makes it into ATP. It's just like water flowing through a dam and turning a rotor to make energy. All right, so it's just flowing downhill. But what if the river stops? Then the rotor stops. right? So you can look at this. If the H pluses are the same on each side, then the rotor is going to stop. So how do we keep this concentration gradient? We got to get rid of the H pluses when they get in here. We also have to get rid of the electrons at the end of the chain. Luckily for us, when we breathe in oxygen, we can take those electrons from the end of the chain. Oxygen is a highly electronegative element. That means it wants electrons. And we can take the H plus protons, combine them together, and make water. Pretty cool, right? This takes some practice. Students, I would recommend sitting down and drawing this out and trying to explain it yourself. It's a pretty cool principle. All right. What happens without oxygen? Well, this is a picture from your lab that we did today. And you can see that it did some fermentation and it made a lot of bubbles. You all did a great job, by the way. So let's go over it. Without oxygen, we can still do some type of respiration, but it's going to be anaerobic. Anaerobic means without. A means without. So here we have lactic acid fermentation. Hopefully you can explain this. Why don't you pause the video? All right, this is going on in humans and other animals and other organisms. We're going to take glucose. We're still going to do glycolysis, and we're going to make 2 ATP that way. But we're going to run out of empty taxicabs. We're going to run out of NAD pluses. We don't have any oxygen, so we know we can't do the electron transport chain because the electron transport chain uses oxygens to accept those electrons and the hydrogens. We also can't do the Krebs cycle because there won't be enough empty taxicabs formed. There won't be any NAD pluses for the Krebs cycle. We can do glycolysis because we can regenerate these NAD pluses in the cytoplasm by doing a little trick. And here it is. We take glucose, we turn it into pyruvate. We use an enzyme to take the high energy electrons and the H plus ion from NADH, add it to the pyruvate, and we'll make lactic acid. That lactic acid enabled us to have an empty taxicab right here. So because we're able to take the occupants of the taxicab and jam them onto pyruvate, we can make lactic acid. We're going to start to feel it burn in our muscles. So then we're going to create an oxygen debt, and we're going to need to pause our exercise and breathe. Here's what you did in class. Okay, you took sucrose, and then it was broken down into glucose with the enzyme sucrase. And in here, these are 2 enzymes. You know who they are because they end in ASC, pyruvate decarboxylase and alcohol dehydrogenase, and it's going to make ethanol. So this makes alcohol. This happens in yeast and in some bacteria. And it's also going to produce carbon dioxide. What's the important point is it's still making ATP for itself, and it's still emptying the taxicab. All right, it's taking the NADH and making it back into NAD+. It's pretty cool when you think about this evolutionarily, which we'll get to in our unit on evolution. All right, students, why is all this stuff important? Well, we'll discuss in class creatine supplements. We'll discuss the poison cyanide, mitochondrial DNA. And last but not least, if you're thinking about becoming a doctor or nurse or anything in the medical field, there's a lot of mitochondrial diseases. And I'll put this in the YouTube video links, but I hope you'll kind of research some of these and see what's happening, what's going wrong in the mitochondria, and maybe how somebody like you can fix it. All right, students, thanks for watching my video. Take care. Good luck studying, and take care.